about chilies, its heterosis and botanical description. Chili is also called as hot pepper, that is Capsicum annuum, commonly known as chili in India, mostly grown in the tropical and subtropical regions of the world as an important vegetable and condiment crop. In India, it was introduced and believed through Portuguese in 16th century. The pungency, red color and oleoresin content in chili are especially important for processed food as well as for export purpose. Capsicum annuum, by far the most widespread species. Origin of chili or pepper. It originated in South America, mainly in Mexico, and spread into New World tropics before subsequent introduction to Asia and Africa. Chilies are now widely grown throughout the tropics, subtropics and warmer temperate region of the world. Taxonomy. Division is Spermatophyta, class Angiospermae, subclass Dicotyledonnae, order Solanales, family Solanaceae, genus Capsicum and species Annuum. Different species of capsicum. It consists of approximately 20 to 27 species. 5 of which are domesticated are Capsicum annuum, baccatum, chinense, frutescens and pubescens. Fruits of capsicum can vary tremendously in color, shape, size, both between and within species, which has led to confusion over the relationships between taxa. Here you can see some of the pictures of different kinds of capsicum. This is Capsicum annuum, which is generally consumed in green form or red dried powdered form. This is chinensis, the wild type. Frutescens, these are having quite more pungency, which are the important source for medicinal or pharmaceutical companies. Baccatum, this is also quite pungent one, or some other subspecies are less pungent type. The inflorescence of Capsicum annuum. The flower diagram, it is a hermaphrodite flower and self-pollinated. Capsicum annuum starts flowering at the axil and first branching node, with subsequent flowers forming each additional node. Usually, Capsicum annuum has a solitary flower at the axil. Flowers are complete with a calyx, corolla and male and female sex organ. It is bisexual, hypogynous and usually pentamerous. The calyx is broadly campanulate, ribbed, around 2 mm long and truncate or undulate to weakly or prominently dentate with 5 to 7 teeth, with usually 5 but sometimes 6 to 7 petals in some species. Typically, flowers have 5 stamens. Pistil comprises of ovary of 2 to 3 carpels with 2 to 5 mm long and 1.5 to 5 mm in dia. The style is 3.5 to 6.5 mm long and capitate stigma slightly wider than the style. The nectary appears as swellings on the basal part of the ovary. The nectariferous cells are smaller and denser than the neighboring parenchyma. Stomata are present in the nectary epidermis but do not appear on the other parts of the ovary epidermis. Anthesis. It starts apparently controlled by day length. The corolla typically opens within the first 3 hours after sunrise, and the petals remain open for less than a day, and there also can be a smaller peak of anthers in the afternoon. Anthesis occur late in the morning between 10 a.m. to up to afternoon, depending on the environmental condition and variety. Period of receptivity of the stigma is 5 to 8 days, from several days before anthesis to fewer days afterwards, with maximum fertility on the day of anthesis is expected. Pollen and fertilization. Pollen grains are medium to light yellow, subspheroidal, pitted and tricolporate with longitudinal grooves. Plant has about 1 to 1.5 mg of pollen per flower, with 11,000 to 18,000 pollen grains in a single anther. Pollen tube growth from the stigma to the egg has been reported to take 6 to 42 hours. Male sterility in pepper was first documented during 1950s. It is considered as knowledge has been accumulated on the nature of trait, means its identification and induction, inheritance of both genetic and cytoplasmic genetic male sterility, its line maintenance and potential for breeding hybrid cultivars. Today, several internationally known seed companies use the genetic mechanisms, that is small ms on a large scale for production of hybrids, whereas cytoplasmic genetic source is used mainly for breeding pungent kind hybrids, that is capital RF and small rf. CGMS was reported by Peterson in 1958 in Chile. Not exploited by public and private sector reasons, hand emasculation and pollination is practicable and feasible in Chile. Then large number of seed set or fruits are crossing. CMS limit no number of lines if they are not complete restorers. Non-CMS based hybrids ensure greater genetic diversity, stable and sustainable production, and finally CMS is only ease of emasculation. The fruit of chili, extensive diversity in fruit shape, size, wall thickness and freshness, color and pungency are found in fruit. Blocky bell-shaped globose, oblong, ovoid, conical, cylindrical, banana-like, smooth, grooved, lumpy or wrinkled, morphologically different capsicum fruit are found, and it is commonly called as berry. Green stage is 35 to 50 days after the flower or pollination. The major problems in chili, one of the problem is chili thrips. Diseases, major diseases, chili anthracnose, generally reported as most serious disease and grown under hot humid conditions. 10 to 85 loss per pre-harvest is observed if it is emerging before the harvest. If it is in the after harvest condition or post harvest, means 10 to 32% loss is expected. Damage caused after all crop production costs incurred. Chili wilt. This is a most dangerous disease in the field condition which severely damage the field. Importance of chili. Chilies contain health benefiting alkaloid compound in them, that is capsaicin, which has antibacterial, anti-carcinogenic, analgesic and anti-diabetic properties. It also found to reduce cholesterol levels in obese individuals. Fresh chili peppers, red or green, are rich source of vitamin C. Vitamin C is a potent water-soluble antioxidant. Chilies contain good amount of minerals like potassium, manganese, iron and magnesium. These are also good in B complex group of vitamins like niacin, pyridoxine, riboflavin and thiamine. Fresh raw bell peppers and sweet pepper varieties are being used as vegetables in cuisines in many parts of the world. Average nutritive in chili. The energy in chili is in kilocalories, 116 kilocalorie. Protein is 1.6 grams. Calcium is 86 mg. Iron 3.6 mg. Thiamine is 0.37 mg. Riboflavin 0.51 mg. Niacin 2.5 mg. Vitamin C 96 mg, and average nutritive value is 28%. Pollination habit. It is generally self-pollinated, but some cross-pollination can occur between and within the cultivars of two species, that is Capsicum annuum and Capsicum frutescens. Anthesis takes place sometime after the flowers have opened. Flower remains open for 2 to 3 days, and it opens between morning 2 to 10 am. 2 am to 10 am. The anthers normally dehisce an hour after flower opening. Flower opening and anther dehiscence to a large extent depend on weather condition. During cold as well as cloudy days, opening is delayed. In sweet pepper, anthesis commences from 7 am, which continues till 11 am, with peak between 7 to 10 am, and anther dehiscence after 30 minutes of the anthesis. The stigma became receptive up to 2 days after anthesis. Extent of cross-pollination. In pepper, frequency of cross-pollination is greater than for tomato. A factor that affects the percentage of cross-pollination that the relative position of stigma and anthers, which depends on the genotype. The presence of pollinating insects and receptivity of stigma 2 to 3 days before pollen shed in some cultivars is another factor. Hand-pollinated seed production. Most of the seeds of F1 hybrid vegetables are produced by hand pollination. The method in principle is simple, as involve the manual emasculation of pollen producing organ, the anthers, followed by hand pollination with pollen of male parent, and then preventing other pollen from contaminating the pollinated flowers. However, it is labor intensive and requires a team of skillful growers. To be cost effective, this system only works in species where a single pollination of female flower will produce many seeds. The length of style and relative position of stigma and anthers vary among genotypes, and it is an important factor determining the level of natural cross-pollinations of the flowers. Steps in emasculation and pollination. These are the techniques in hybrid seed production. We should, as the other crops are solanaceous crops, we have to select a bud which is unopened and will be expected to open within a day. And after selecting it, this is a suitable bud which is ready to be emasculated, and after that we have to remove the anthers like this. After removing, we should expose the stigmatic portion, and after that covering a bag. Next day we have to pollinate and cover and label with a suitable label consisting all the information of parentage and date of pollination. Male sterile lines. Hand emasculation and pollination are most expensive methods in hybrid seed production because of high labor cost and low seed set. Genetic male sterility is most commercial and exploited for hybrid seed production. This I already explained in the previous slides, but again I will tell you about this. The male sterility in pepper was reported first time by Martin and Groford in 1951. Since then, male sterile genotypes in this species have been obtained by mutagenic induction or by interspecific hybridization. Much information information has been published concerning the nature, inheritance, and stability of pepper male sterility and the possibility to include sterile forms in breeding program and sterile lines in hybrid seed production. Genetic male sterility. This is generally controlled by a single recessive gene, that is MS, and has been reported in tomato, brinjal, chili, sweet pepper, pea, lima bean, pumpkin, squash, watermelon, muskmelon, ridge gourd, cucumber, cauliflower, cabbage, broccoli, and Brussels sprout. The male sterile line needs to be maintained by crossing to a maintainer line heterozygous for recessive male sterile alleles. Thus, 50% of the progeny will also be heterozygous and fertile. Therefore, that should be rogued out regularly before flowering, which is a problem again in hybrid seed production, as the hand emasculation and pollination are labor intensive, but genetic male sterility, it's a waste of energy where we have to remove the 50% of plants. The efficiency of this male sterility GMS system can be increased by using closely linked marker gene so that the male sterile plants can be detected prior to anthesis, preferably at seedling stage. This technique so far has not got wider acceptance and commercialization. CMS or CGMS. CGMS was reported by Peterson in 1958 with introduction of Capsicum annuum from India, PI 164835. It is unstable under fluctuating environmental condition. By carrying out cytological characterization, two sterile lines with recessive gene MS3 and MS8 and one CMS line, that is S by MS-5 MS-5, was obtained by Discolo. These are the parental lines, and after crossing, the F1 was having more yield as well as the attractive appearance as compared to their parents. There are certain reports I have given here about the heterosis and other aspects of chili for your reference. Heterosis for various attributes in hot pepper. For different traits, different cross combinations are better, and that is also given here. These are the case studies. Nationally released hybrids of chili. ACH15, ACH30, these both were released from Gujarat Agriculture University, that is Anand. Then from Bijo Sheetal Seeds Private Limited released BSS 141 and BSS 138 were the popular one. From PAU Ludhiana, CH1 and CH3. CH1 is a hybrid developed by MS12 into LSS, a popular hybrid. It is medium tall with light green foliage. Fruits are of light green when immature but turn deep red when shining surface at maturity. Due to its high pungency, that is 0.83%, and red color, its fruits get premium price. Dry matter content is 23.8%. It tolerant to diseases like fruit and wet rot, viruses like mosaic and leaf curl. Low incidence of fruit borer and sun scald were recorded. Its fruits are highly suitable for drying and used as salad. Yield of chili is 250 quintals per hectare, but in case of fruits are picked at green stage, the yield is almost double, means around 500 quintals per hectare in green form. CH3, again it is released from PAU Ludhiana, developed by cross between MS12 into 2530. It's medium tall, vigorous with dark green foliage. Fruit length and fruit weight is higher than CH1. Immature fruits are dark green but turn dark red at maturity. It is about 7 to 12 days early maturing than CH1, and color is also attractive, tolerant to different diseases more than CH1, and yield is 275 quintals in dry form or red chilli form. Hybrids from a private company, and the list is given here. Delhi Hot Skyline Hot Green from Hong Kong company. Then Mahiko released Tejaswini Sujata Surya. Novartis released Agni, Bichli, Picadas. Then Ankur Seeds released ARCH series, Chilli. Then some other companies are also given here.